Antibodies and Disease Part 3, Making Antibody Drugs and What My Group Does to Help. Producing antibodies. So, the traditional way of producing antibodies is to take an antigen of interest, inject it into a mouse. The mouse then produces B cells which express antibodies against the antigen. You fuse those B cells with myeloma cells to produce so-called hybridomas, which are then immortal and go on producing the antibody of interest. However, we can't do this with human monoclonals. We can't produce human antibodies in the same way that we can produce mouse antibodies. There are a number of reasons for that. First of all, if you're trying to produce an antibody against a human protein, then injecting that human protein into a human won't work. Your immune system is set up not to produce self antibodies that will bind to self proteins. Secondly, if you were interested in an exogenous antigen, something like a toxin, you couldn't go around injecting toxins into people to produce antibodies against them. So instead, if you're producing an antibody in a mouse and you take that mouse antibody and you inject that into a human, then the human will recognize that as being a foreign protein and produce an immune response against that. So using mouse antibodies doesn't really work either. Now, back in 1984, Morrison et al. Started to approach this problem by producing what they called chimeric antibodies. In a chimeric antibody, you're taking your mouse, or it might be a rat or some other species, you're taking your antibody from that, and in the chimeric you're using just the variable domains from that antibody, and you're fusing them with constant domains from a human antibody. Now, of course, you're doing that at the DNA level, not at the protein level. And that has two advantages. First of all, the variable domains are the only parts that come from the foreign species, and therefore you're reducing the chance that this will produce an immune response. And secondly, the Fc fragment here is a human Fc fragment, and if what you're requiring is that this interacts with the rest of the immune system, having the human Fc is going to work better than having a mouse or rat or whatever Fc. However, in the chimeric antibodies, there is still a substantial amount of foreign protein present. So the refinement to that then is known as humanization. Now, this is a view you've seen before of the antibody combining site, and we have the 6 CDR loops shown in the purple, orange, yellow, green, red and cyan. And the rest is the supporting framework. Now, given that these 6 CDR loops essentially form the combining site for interacting with the antigen, then humanization takes those from the foreign species, like mouse or rat or whatever, and the rest of the framework, as well as all the other domains, the constant domains, from a human. So this was first done for a complete antibody where you're looking at both light and heavy chains and where the antibody was binding to a protein. It was first done by Reichman et al. In 1988 in a paper that was published in Nature, where they took the CDRs from rat and grafted them onto human frameworks. Now, what they found when they did that was that the antibody didn't bind very well. And so they had to look to see why that was happening. Now, if you recall, when I was talking about the detailed structure of CDRs. We have these key residues in the CDRs themselves, plus residues that interact with the CDRs from the surrounding framework. And together, these define the overall conformation of a loop. Now, given that we know that that happens with these interacting residues in the framework, what Reichman et al., working in Greg Winter's group in Cambridge, the same place that Cyrus Choudhury was working on the canonical conformations of the CDRs, they realised that interacting residues might have an effect on the fine, precise structure of the CDRs. So in this particular instance, this in red is CDRH1, and there's a tyrosine at this position. Now, in this particular rat antibody, the tyrosine was interacting with a phenylalanine at this position, H27. And that's shown in orange. Now, the human framework that they decided to use had a serine at that position instead of a phenylalanine. So a much shorter amino acid. It would stop at this point here with an oxygen. And they found that mutating that serine to a phenylalanine restored the binding. So this is just one particular example. This happened to be a serine to phenylalanine that was needed. This is not a universal solution. It's just a position that was interacting, and they needed to make that change in order to get the interactions correct and maintain the conformation of this CDR. They also had a look at position H30, so that's this position here, where there was a serine in the human framework, but there was a threonine in the rat. This is outside the CDR, but they thought because it's right on the surface, right next to the CDR, that it might still interact with the antigen. They tried making this change, and it made virtually no difference. Now, of course, this is commercially a very interesting area. Trying to find which residues you need to change in order to recover really good binding has a lot of value as a technique. And that means that there are a number of patents that have been issued in this area. One of the earliest being the Adair patent from Celtec, that then became UCB Celtec. And the first claim of that patent refers to these four particular residues here. H6, which is this one, which, okay, it doesn't interact with the CDRs, but it packs straight in towards the framework that's supporting the CDRs. H24 at this position packs inwards against the base of some of the CDRs, and H49 is right next to the base of CDRH2. Now this 123, it's not really obvious how that could affect either the conformation of the CDRs, because it's pointing outwards, or the interactions directly with the antigen, which is up here. But it actually can make quite a significant difference when there's a very different residue here. In the patent, it was looking at a difference between a serine at this position and a lysine. And this residue, it seems, interacts with this extra loop that I mentioned earlier, alters its conformation, and that in turn interacts with the CDRs. Now, I said that we can't produce human antibodies, and still an awful lot of antibodies are produced by the techniques that I have talked about, producing these humanized antibodies, these CDR grafted antibodies. However, there are an awful lot of antibodies being produced these days that are fully human. Now, one technique for doing that is this method called phage display, in which we produce a library of bacteriophage. So bacteriophage are little viruses that infect bacteria. And using this idea of single chain FVs, where we have the VH and the VL linked together, we can actually link those on to one of the coat proteins on the surface of a phage. So here we've got 3 phage, each with an antibody stuck onto it. So what we have then is this library of these phage. We fix our antigen of interest onto a plate, and then we add the phage, and some of the phage will stick. So then we wash off the ones that don't stick, we elute the ones that did, and we go through a phase of stocking and titrating what came out. We can then go through an amplification stage and run round this cycle 3 or 4 times, and by doing that we end up with a set of antibodies that bind really well to the antigen of interest. Now, during this amplification stage, we can use low stringency DNA copying, which means that some errors can get introduced. And by doing that, you can actually simulate the sort of thing that goes on in the immune system of this somatic hypermutation, where additional mutations get introduced. So that's one technique that has been used. Another technique is to use transgenic mice. What we do in these transgenic mice is to modify mouse embryonic stem cells such that we introduce antibody genes from humans. We also produce mice that have both their heavy and light chain loci inactivated. And then by breeding these two together, one which has both mouse and human antibodies and one which has neither, we can end up with just mice containing just the human antibody genes. We can then challenge these transgenic mice with the antigen of interest. The mice start to produce B cells which are producing human antibodies. So we can then go ahead through our usual route of immortalizing these B cells to produce hybridomas and then secrete and purify fully human antibodies. And once we've got the sequences of the antibodies by any of these methods, we can then produce them by expression in things like CHO cells, Chinese hamster ovary cells, or HEK cells, human embryonic kidney cells. So where do we come into all of this? Well, largely through our development of a resource called Abyssis, which is available for people to use publicly over the internet, but which we also sell to companies to have in-house. And this is a big database of antibody sequences and structures, together with a suite of analysis tools that allows you to look at antibody sequences and compare them with these resources of sequence data. So one of the things that we have as part of that is a nice kind of alignment viewer, which allows you to select a region such as a CDR. So the CDRs here are highlighted with this little red stripe. So you can select just a region like that, and you can sort the sequences based on those regions. You can then go through by clicking on these links to look at the sequences in detail. You can hide them and so on. If you look at a sequence in detail, you get information like this, which includes frequency information. So in this case we're comparing our particular sequence against all other organisms, but methionine at this particular position is seen in fewer than 1% of other sequences. This site, as it says down here, is also a methionine oxidation site. So you can look for things like unusual amino acids, which, even if it's a fully human antibody, may lead to an immune response against that antibody. You can look for features where post-translational modifications may occur, like these methionine oxidation sites, or in this case, a CK2 phosphorylation site, in this case, a PKC phosphorylation site, and so on. And these are all things that can impact on the developability of an antibody as a drug. In this instance, we're looking at this particular position H1. So if you clicked on that, you would get a graph like this and a table like this showing you the frequencies of residues at that position. Now, what we have here is an E, a glutamate, at position H1, and you can see that that's a common residue together with Q, which is glutamine. Over on the right here, we have an assessment of what we call humanness. So I don't have time to go into a full explanation of how humanness is calculated, but essentially it's a way of taking a sequence and comparing it against all of the other antibody sequences to see how representative it is of the human repertoire. So the little graph in the solid lines is a distribution of what human sequences look like, while the grey line is a distribution of what mouse sequences look like. Now, this particular sequence is a mouse one, and that's indicated by the dashed line here. So you can see that this is really sort of towards the peak of what mouse sequences look like, but it's at the low end of what human sequences look like. Again, this potentially is linked to how likely a sequence is to generate an immune response against it. If the crystal structure of the antibody is known, then it comes up with an interactive structure viewer where you can rotate this, you can modify the colouring, and so on. What we have just added into the system at the moment is a link to an antibody modelling program. So if there is no structure available for your particular antibody of interest, then it can go away and generate a structure, and this will then come back in this tab just as it did before. Given that we have this system where when an antibody is created, it's this merging of a V segment and a J segment for the light chain variable regions, or V, D and J for heavy chain variable regions, but this then undergoes somatic hypermutation, it's often of interest to know which germline gene, which original gene, this antibody came from. So we have a system here that allows you to do that. You put in your sequence, which can be a protein sequence or a DNA sequence, and it will go back and find the gene segments that this was most likely to have been derived from. Now, this is an extension of what we saw with residue frequencies. So you might recall that with that sequence that we looked at, position H1 was, in this case, a glutamate, E, but the other common residue at that position was glutamine, or Q. So what we have here is a heat map of that same information. So at position H1, you can see we have a green blob for glutamate and glutamine, and all the other ones were unusual, so those are all red. And the heat map actually allows you to define two different thresholds, which you set down here, so that common residues are shown in green, uncommon residues are shown in red, and something in between is shown in yellow, and you can decide what thresholds you want there. So overall, the system that we have developed allows people to look for features in their potential drugs that might be bad from the point of view of being unusual, to make this sequence something that is not typically what's seen in human antibodies, and therefore may be recognized by the human immune system as being not a natural protein, and therefore the immune system would attack this antibody-based drug. We also allow the user to look for these features that might lead to post-translational modifications, in turn leading to instability, changes to the nature of the protein, which would make it less effective as a drug, either during storage or once it's given to the patient. So I'm going to end at that point. Thank you for listening, and please think up any questions that you have to address in the question and answer session.